Hello, everybody. I hope you are having a great day. Welcome to my YouTube channel. On this channel, I give you all the tips and tricks and resources that are really beneficial in my biology classroom, and I show you how to recreate them for your classroom. So today, I am going to show you how to create a protein synthesis activity that involves real human genes and how to use a government database in order to create the DNA sequence from the gene sequence. The gene sequence you'll find out is an actual amino acid sequence. So we have to transcribe and translate backwards. And then what you give the students, you'll give them the DNA sequence, and then they will transcribe and translate that sequence. Then they will go back to the same government database that you used, except this time they will input their amino acid sequence and hopefully come up with their gene as long as they transcribed and translated correctly. It's a really fun activity. My students love doing it, and I love it because it sort of self grades. Once they transcribe and translate, they go into the database. If their gene pops up, they did everything correctly, perfect, you get a 100. If the gene does not pop up, then they have two options. One is they can go back and check the work on their own, or the second option is they can ask me to check their work, but if I check their work, they lose 15 points. So it's still pretty easy, even just going back and checking their work because you'll have the master. And most of the time, the students prefer to go back and check the work themselves, which is great. So let's get started, and I'm going to show you exactly how I create these sequences. And all of the websites that I show you in this video, I'm going to link down below so you do not have to go and search for them. You can just click the links. Okay, so to get started, the first website you're going to start on, everything we do, by the way, is on this NCBI website from the NIH. So the first page in the website is on genes and diseases. And what you want to do here is to go through this list of diseases and pick the genes that you want to use. In the activity that I gave my students, I use 6 genes. You can pick as many or as few as you want. So I'm just going to show you how I find the genes. So I'm going to click cancers, scroll down, breast and ovarian cancer. Try to pick diseases that are very well known, that we know there's been research into the genes, because it's just going to make your life easier, okay? You'll see once we get further into this. I'm going to pick BRCA2, so BRCA2. Now, you can either highlight it or you can just remember it, but you are going to go to the next website, okay, right here. Same website, still NIH, different page. And again, I'm going to link all of these websites so you do not have to go and try to find them. It's a nucleotide search. So up here, you start typing in your gene. So I want BRCA2. Make sure Homo sapiens is there. So I want Homo sapiens BRCA2. And I want the full genome. So I'm going to add full. I found if you don't add full, a lot of the times they give you truncated and partial genomes, and it's just not what you're looking for. So then you scroll down, and I already did this, so it's going to pop up, but you can see right here, the first one, this is the partial CDS. You don't want that. You want something that says complete. Even though it's truncated, that's okay. There's still 5,220 base pairs. This is going to give you what you want. I'm going to show you exactly what we're looking for. So you scroll down, do not worry about any of this. This is what you want, where it says translation, because this is the information that you want your students to actually end up with. Now, when I do this, I pick approximately 18 amino acid letters. So you're working with 18 amino acids. I have found that it's a short enough version where the blast will actually work. Okay, so I just selected a bunch. There's approximately 18. I hit Control C. Next website, still on the NIH. Again, I'm going to link everything. This is a BLAST, so a basic local alignment search tool. What that means is we are going to input the amino acid sequence, and hopefully this will work. We'll get our gene back. What happens is with the students, once they transcribe and translate their sequence, this is how they are going to find out whether or not they transcribed and translated correctly. So you give them this website, you want the protein blast. So you click on it. You just paste your amino acid sequence. You scroll down and you hit the blast button. Now, this portion of the activity can take anywhere between 1 and 7 minutes. I've been in a classroom before where it's taken a student 10 minutes. I tell them patience is something that they have to practice. What will end up popping up, because I did this earlier so we don't have to wait 5 minutes on the camera, this is what the screen will look like. Students will scroll down, and here you go. You have your BRCA2 gene. When you are doing this, what you are looking for is, number one, you have to find the sequence. Number two, when you put the sequence into the blast, you have to make sure that the actual gene comes up. If I did not pick 18 amino acids, and let's say I only chose 5 and put it in here, it could be some random genes that pop up. And that has happened to me before. And I've even input 18 and I'm still not getting the gene. So I just go back to the drawing board and I just find a different gene. You want to make sure that the amino acid sequence works and it actually links to the gene that you want the student searching for. So now that you know it works, you go back to this amino acid sequence. I'm just going to highlight it again. Okay, Command C. So I'm just going to copy it. I open up a document. I prefer to work with Google Slides. I find it easier than Google Docs. And then I just paste the sequence. That is very hard to see, so I'm going to change it. There we go. So this is the amino acids. Now, what you have to do is you have to work backwards because you want to give the students a DNA sequence, not an amino acid sequence. So from the amino acid sequence, you have to figure out what the mRNA is, and then from there, the DNA. You do this by opening up an amino acid codon wheel. So I have one right here. I just downloaded this one. And you start letter by letter. So the first letter is M, methionine, and that, as we all probably know, is AUG. So here's methionine, the M, AUG. So I go here for mRNA, I will type in AUG. I'm not going to do all of these with you. I'll just go through 2, okay, just to give you an idea how you do this. The second letter is P, so you go back to the wheel, you find the letter P, it is proline. Now this is where you just pick. You're not giving the students an actual DNA sequence because that I have tried to find the actual DNA sequences. It is just way too difficult. It's much easier to find the 1-letter abbreviated amino acid sequence and just reverse engineer it to create the DNA sequence. So when I get to something like proline where you have 4 different codons that you can choose from, just pick one, okay? Just pick. So for proline, I'm just going to pick CCC. Then, in order to create the DNA sequence that you are going to give to your actual students, you have to, again, reverse engineer. So you have to create the DNA sequence from the mRNA sequence. So AUG would be TAC, and then CCC is GGG. Now you have to do this for the entire amino acid sequence for as many sequences as you want to assign the students. They will get the DNA sequence. Let me show you what my final product looks like, what I hand to the students. So this is my sequence number 5. Let me zoom in a little bit so you can see it. So sequence number 5, again, I give my students 6 different sequences. The title of mine is It's in Your Genes. I give them directions. They have to transcribe and translate to identify which gene sequence you have been given. So here is the DNA sequence that I reverse engineered from that single letter amino acid sequence. They have to give me the mRNA sequence, then the amino acid sequence, and then the single letter amino acid sequence. You can combine these 2 and give them a wheel like this and say, just give me the single letter. That is up to you. I like them to do the extra step. Then, so page 2, I show them and I give them directions for what they are going to do. So what we did in the second half, when we went to the blast, if I hit back and back again. So this web page, the protein blast, this is where they're going to start on the website. So if we go back to here and I screenshot it, I circle it, I make an arrow, like this is where you click, guys. And then I tell them where they're going to enter their single letter amino acid sequence. It does not matter if they use capital or lowercase letters. Just tell them not to mix them. Pick capital, pick lowercase, don't mix them. Then click the blast, wait some time, and then they scroll down. So these are the genes that I chose. If you wanted to use these, go right ahead. And then you can see, like I showed you how to use it and get the results for the BRCA2 gene. This is the MSH2 gene. So MSH2 just comes up over and over again. So if they had sequence number 5, they get to the end, they see their gene pop up, that is great. For all of my sequences, they have this same directional sheet. So it's not like, oh, if you have sequence 5, you are going to get MSH2. I did not tell them that part. This is just like, hey, this is what you should get. This is what you're looking for. Now, I take it a step further. After they finish the blast, if they get the correct answer, then they go to the internet, they figure out they write what their gene is first, which they found from the blast. Then they go to Google, they type in this sentence in order to figure out the disease that their gene is associated with. All of the genes here that I use works with this Google phrase. And then it comes up with the disease. And then I ask them to answer these 3 questions about the disease: the description, symptoms, and treatments. But that is up to you. And this is how you create a protein synthesis activity that uses real human genes, the amino acid sequences. You just have to reverse engineer the sequence to get the mRNA and the DNA, and then you give the students the DNA. If you want to do this on your own, great. If you want this version that is already made with answer keys, just click the link below. I will link it. It's up on Teachers Pay Teachers. I think it's $3. It's not that expensive. I hope you found this helpful. If you did this and you did the genes, you created your own sequence, you used it with your students. If it was great, if your students liked it, let me know. If you have any questions at all when you're using the database or trying to do this, make a comment, send me an email. I'll be happy to help you out. Take care, guys.